Hi everyone, my name is William Zeiger, and I'm an assistant professor of neurology and a movement disorder specialist at the University of California, Los Angeles. Parkinson's disease is one of the most common disorders that I encounter in my clinical practice. When I work with patients with Parkinson's, we spend much of our visits talking about how to manage their movement symptoms, things like muscle stiffness, slowness, impaired dexterity, and walking problems. Fortunately, we have a lot of good treatments to manage those symptoms. On the other hand, many of my patients and persons with Parkinson's disease in general suffer from additional non-motor symptoms. These can be things like speech impairments, bowel and bladder problems, blood pressure issues, or mood disorders. Often, these are just as troublesome as the movement symptoms. And among these non-motor symptoms of Parkinson's disease, one of the most feared and potentially most debilitating is cognitive impairment. We know from natural history studies like this one that many persons with Parkinson's will develop thinking and memory problems. Problems with attention, concentration, planning, and multitasking are very common in persons with Parkinson's disease. Difficulties processing visual information or memory impairments can happen as well. And for some people, these problems become so significant that it makes it hard for them to function independently, a condition we call dementia. Unfortunately, we lack really good treatments for these thinking and memory problems, and we don't have any way to slow down, stop, or reverse this process. Studies over many years have helped us understand that certain parts of the brain are involved in thinking and memory and are particularly susceptible in persons with Parkinson's disease. For example, on this picture of a brain, the orange and yellow colors indicate areas toward the back part of the cerebral cortex that show thinning and atrophy in persons with Parkinson's disease. The brain circuits in these areas help us process visual information. And we know that persons with Parkinson's disease, dementia, have difficulty processing visual information. We can test for these problems in the clinic in a variety of ways. In this example, you can see some white dots moving around a black background. When most of the dots are moving in the same way, it is easy to perceive the overall direction of motion. But when many of the dots are moving randomly, and only a small portion are moving in the same way, it gets much harder to perceive the direction of motion. Persons with Parkinson's disease dementia have particular difficulty with this task, whereas persons with Alzheimer's disease or persons with Parkinson's disease without dementia can perform well. In our laboratory, we're interested in figuring out exactly how Parkinson's disease causes dysfunction of brain circuits, and how that leads to the specific cognitive problems that our patients experience. It is really hard to study this in humans. We can't routinely peer into our brains and see what's going on. But fortunately, we can turn to mice for help doing just that. In fact, we can teach mice to perform these same visual processing tasks that our patients struggle with. We can then model Parkinson's disease in our mice. In this example, we induce the same kinds of pathology that affects the brains of persons with Parkinson's disease, here shown as green clumps of a protein called alpha-synuclein in the brain of a mouse. Finally, we can use a specialized type of microscopy to record the activity of individual cells inside the brain of a living mouse. All of these little circles are individual cells in the brain of a mouse. The flashes that we observe represent the activity of these brain cells as a mouse views and processes visual information. Together, these tools help us understand how Parkinson's disease affects the function of brain cells and brain circuits, and how this might lead to thinking and memory problems. With the help of the American Parkinson's Disease Association, we are now able to ask these questions in even more detail. The brain is made up of many different types of brain cells, all with unique functions. We can use genetic techniques to actually look at the activity of specific genetically defined subpopulations of brain cells. In these pictures, we can see a type of inhibitory brain cell called parvalbumin interneurons. We can actually follow the same exact cells over time and see how their activity and function changes as Parkinson's disease pathology progresses. In this way, we can understand how different types of brain cells work together and how Parkinson's disease might cause problems in certain types of brain cells more than others. In summary, we're really honored to have the support of the APDA, which allows us to pursue this important research. Our research will help us to understand how thinking and memory problems arise in persons with Parkinson's disease. Going even further, it will help us understand how specific brain cells and circuits are affected. And finally, it gives us a model in which we can test new strategies to slow down, stop, or maybe even reverse thinking and memory problems in persons with Parkinson's disease.